I'm pleased to welcome everyone to the second session of our series today. Our overall series is called An Introduction to Biologic GMP Manufacturing, Preparing for Early Phase Clinical Trials. And so this is a joint effort on behalf of weCANreg, Stem Cell Network, and the NRC. I'm Megan Mahoney. I'm with weCANreg, and I'm the Director of Scientific Affairs and Training Programs, and I'm really happy to be facilitating today's session. So to begin, I'd like to acknowledge the land on which we are gathered, which is also the land on which I live. We're gathered on the unceded territory of the Anishinaabe Algonquin peoples. These peoples have lived on this territory for millennia. Their culture, presence, and contributions have nurtured and continue to nurture the land on which we are operating and live on. I acknowledge and respect them as traditional hosts and for allowing us to operate on these lands. So last week you heard about Stem Cell Network. Today I'd like to tell you a little bit more about weCANreg. We are a federally funded, not-for-profit entity in the area of cancer immunotherapy. Our vision is to turn all cancers into curable diseases, and we do so, or we are trying to do so, by bringing together a network of clinicians, scientists, cancer stakeholders, academics, NGOs, and industry partners for the benefit of contributing to cancer immunotherapy. As a leader, we take part in a number of different activities, from research through to training, biomanufacturing, and this is really the impetus for today's session and for our involvement with the NRC and Stem Cell Network. So with that being said, I'll keep that brief, but I do invite you all to take a look at our website, biocanreg.com, and to check us out on social media at biocanreg. So with that being said, in case you missed it, you can check out our last session on the Stem Cell Network's YouTube channel. So there is going to be a post in the chat box momentarily. And with that being said, we'll move on to housekeeping notes. So if you have any questions during today's session, you can post them in the Q&A box that is at the bottom of your screen. And if you have any other technical difficulties, you can write us in the chat box. There will be closed captioning enabled for this session, so you can choose to incorporate that if you wish. And this session will also be recorded, so that will also be found on the Stem Cell Network's YouTube channel. So now we're going to launch some polls. Again, this really helps introduce the panelists to who you are in the audience. So there should be 3 polls that will pop up on your screen momentarily. So there they are. So while those are being answered by all of you, we'll move on to the highlights of today's session. So there are a number of points that we hope that you learn and take away from this session. So broadly speaking, the focus will be on effectively coordinating your CMC technical and preclinical operations from a regulatory perspective. You'll gain a better understanding of quality-centered development strategies for scalable processes and robust analytics. That will become clearer later on. Learn about important considerations for the execution of preclinical studies, and finally have a better understanding of core CMC milestones for supporting your early phase clinical filings. So with that being said, I think, Gustavo, let me know when the polls are ready to close and we can take a look at the responses. Perfect. So I think similarly, like last week, we see the majority of folks are coming from academia, but we do have representation from the private sector, as well as a few from government and other. In terms of how confident folks are in terms of transitioning your product to clinical grade GMP manufacturer, most people are neutral. We have about equal representation of both confident and non-confident, and a few that are very confident. So that's good. I think we'll try to address as many of those and really try to push folks into the confident, if not the very confident range. In terms of biologic type, the vast majority is cell and gene therapies with a various smattering of other monoclonal antibodies and vaccine and therapeutic viruses. So that generally, I think, reflects the audience that we're going or that we are targeting. So that's always good to see. And so now I'd like to introduce our panelists and really thank them for being there, and also for everyone that has helped with the working group and developing of this session from the Stem Cell Network and also from the NRC. But to introduce our panelists, we have two panelists today. One of their colleagues will join us for the Q&A and the panel discussion near the end. But today we've got Patrick Bedford and Panos Chrysanthopoulos, and they are joining us from a newly incorporated organization called weCANdev Consulting Group. So this weCANdev Consulting Group is a member of the weCANtranslate network. Together, our speakers have over 50 years of collective experience directly relevant to supporting cell and gene therapies throughout the different stages of development. And they've contributed, you probably know of them, they've contributed quite a lot to Canadian startup community and are now focused in the cell and gene therapy areas and working with companies to identify key activities to move the needle forward for value and for regulatory approvals. So today we've asked them to talk a little bit about the ways that they and we can work toward developing a product from translation through to clinical trial and beyond with the CMC lens. And they'll go into detail into that. And from there, they'll talk a little bit about the manufacturing preclinical alignment and also how to leverage key development philosophy of quality by design, which I think many folks have heard by this point. So in terms of each of them as individuals, Patrick is very qualified. He's been in the space for quite a long time and brings a regulatory lens, and has written and led a number of teams who wrote the Canada's transplant guidance. He's held the pen on cell therapy clinical trial guidances. He's helped over 50 emerging companies establish and navigate regulations. And he will start off by aligning our manufacturing and preclinical studies before handing it off to Panos. So then Panos is a cell therapy leader with process and analytical expertise. He's the Chief Development Officer at Morphicell, an emerging cell therapy company. He's scaled manufacturing towards GMP for small and large biotech companies. He was a senior leader of CRISPR editing companies' development efforts, and he's going to take us into a deeper dive of developmental perspectives of cell therapy, focusing on key manufacturing activities during translation. So with that being said, both of you, I'll turn it over to you and let you take it from there. Welcome.
Great, thank you very much, Megan. I'm just going to do a quick mic check and make sure you guys can hear me okay.
You sound great. Yeah.
Perfect. And now I'm trying to share my screen. Let's see. It took me out of presenter mode, but hopefully I can fix that quickly. There we go. Can you confirm you can also see my slide deck now and it's full screen?
Looks great.
Perfect. Great. Well, thank you very much for having us to weCANreg and Megan for the introduction, and to Stem Cell Network and to NRC for organizing these sessions and inviting us to participate. I want to start off just by saying I've worked and spoken a lot with these organizations and really enjoy what they do in the Canadian ecosystem. I think it's really important work, and I'm always happy to contribute. So here you see the learning objectives for our hour today, and then our discussion. We are, as Megan said, really going to be focused on how to align some of our manufacturing efforts and process development efforts with preclinical efforts. I'm going to be speaking primarily from a high level and a regulatory perspective before turning it over to Panos, and hopefully together we scratch any itch for why you're here. And thank you very much for coming and for your interest. We had a quick intro, but I wanted to highlight that the weCANtranslate does have a diversity of perspectives, different experiences, different people. We have people with regulatory backgrounds, process and analytical development backgrounds, clinical trial and preclinical study expertise. And we've worked in a combination of industry, government, not-for-profit, and other settings. So that it's not too confusing, I want to just quickly highlight, we are a number of organizations. They're independently incorporated. weCANtrial, weCANdev, and weCANreg, and together we work off of an integrated playbook at weCANtranslate. When I say translate, I feel like I really need to define what I mean. And I mean translate more detailed than some people. Sometimes when we refer to it, we do it in a very nebulous way, and we talk about the significant effort and money that needs to be put into it, and then a little bit of magic. But I'm going to try and be a little bit more specific. So I'm not going to focus on how do we get to regulatory success or cheaper products. These are key outcomes, but they're not the activities that we're going to be highlighting right now, because those aren't sufficiently SMART goals to help us actually move the needle and translate. And I think they lead to a lot of failure if we look at just what the outcome is and not part of the process. Instead, I'm going to simplify at a high level and say translation is really aligning to a plan, and we're going to refer to quality by design. It's getting our manufacturing to good manufacturing practices that's scalable and cost effective. We're going to get to safety in terms of preclinical studies that get to good lab practices. That's the acronym GLP. And that's under specific conditions that are reliable enough using a relevant animal model to convince regulators that we can start trials. And then it's about getting efficacy to meet good clinical practice. So this process and how we do that is actually the magic of translation. And we should always keep an eye on improving this process and making sure we do it more efficiently. That's the only way that we're going to get cost-effective drugs. So today's talk is really going to focus on getting the manufacturing and the preclinical studies and how we align it. And it's after we do the strategy, we're going to be giving the strategy and then showing how this can align in time. So just to provide our basis of knowledge of process and analytical development, I'm relying on the International Council for Harmonization. So this is globally relevant and drawing from ICH Q14 and Q2, talking about analytics, but this is loosely applicable to process development as well. So we have different stages. The first stage, and a lot of you may be at this if you're in academia, predominantly done by academia, is this development work. So this is, you have an idea, you've got some IP around it, now you're advancing, you're using research grade material, and you might be working with proof of concept in vitro and in vivo animal models, and you're doing some rudimentary improvements to your technology, and that's considered the development stage of product development. The next stage that you should have an eye towards, and this is as you advance towards GMP, which is good manufacturing practices, and in a specific establishment that has the quality procedures in place, quality management systems to advance, you should be looking towards getting to good lab practices. So this is trying to use clinical grade material, preclinical GLP, and clinical trials. That's kind of validation. As you know more about your product, you're more able to navigate this, and you're learning about the safety and efficacy implications. So critical quality attributes that potentially affect people. And that's the validation stage. This is often done during first in human and later pivotal studies until you plan to apply this in a commercial scale. So you have scaled manufacturing, clinical studies that are late stage, and getting into market authorization. So keep that in mind. I'm speaking to different groups, and different groups throughout this have different requirements and goals. And I'll go over the key challenges that I hear, having seen a number of companies navigating these throughout time. So here what you see is a basic timeline of commercialization or getting to the market or treating patients regularly, where you start with ideation, product definition, prototyping, often in academia, before doing some tech transfer either to industry or spinning out a company, where you develop a company, develop a product, you validate, test, then you move on towards commercialization. And I've put in the hope hype cycle right here that starts, you get very excited when you have the idea, you encounter some challenges over time. And this typically is when you're realizing the need for developing your analytics and process. And then you realize, ooh, we have a bit of a gulf here. We need a lot of investment to do some more of the science and to start assessing our product in preclinical study models. And that's often referred to as the valley of death, before you realize that data quality and all these other things are required. You have to qualify all of your analytics and eventually validate them all. And then you finally have a realization of your product. So the first first problem that people encounter is usually uncertainty. People refer to this sometimes as regulatory uncertainty. They realize that they can't just keep doing science at the bench side. They have to invest heavily into third-party wet labs. They have to start getting different materials that are more available at a commercial scale. And how to do that is really the first problem, not knowing how to do it. So why should I invest all this money in doing this? Is the first question I get asked. And then the second problem that I think people encounter is they realize the challenges in getting good quality data. So how do you create a culture of collecting data and following SOPs and stopping the innovation cycle with your creative, brilliant scientists and forming a group that's really focused on doing the repeatability assessments and things like that. And then finally, we have this problem of consistency and cost. And unfortunately, there's not much you can do about consistency and cost after your complete product has been developed. It's really hard to do post-market changes. So this problem actually will come back and be identified in later stage product development, but really should be thought about ahead of time. And I don't think all of us know the costs of cell and gene therapies right now. We have a general idea of why they're getting expensive, but really the time to resolve this is much earlier. So now that we know those challenges, from a manufacturing perspective, I'm going to go over similarly 3 stages of preclinical development and then their challenges. So first off, we have proof of concept. And this is my way of thinking about it. This is not, I think, how it's generally taught or broken down. It's just, it's breaking things down into useful components that I find very helpful. I find thinking about the animal model studies, towards GLP, this is that part of commercialization of getting your safety to GLP studies. Think about all of these early studies that people do. A lot of it in Canadian academic centers are for yourself. You're convincing yourself that you have an early product candidate that's worth developing further. And it's part of your narrative of why you selected it. And some of this data may be helpful, but it's not directly relevant to your regulatory submissions often. These are really helping you identify what you'll hear people in quality by design speaking about your quality attributes, your critical quality attributes. You're exploring that at this stage. And then you have these early clinical studies. I'd suggest that these studies are also not going to move the needle directly for regulatory submission. These are de-risking your big investment into using material that's going to go into people, the GMP material, and de-risking your pivotal key safety preclinical studies, making sure that you're getting results that are interpretable from your animal models. And I think the biggest receiver of this data is actually your investors and yourself. So you're de-risking this this path by getting GMP-like, the most relevant data in a relevant animal model, and you're assessing it, and you're looking at efficacy at this time to say, yes, this is truly the candidate that we want to bring into the very expensive GMP manufacturing setting and very expensive GLP preclinical study setting. And then finally, you hit these, what are sometimes referred to as pivotal preclinical studies that are truly focused on key safety issues. So this is the ectopic for cell and gene therapy. This is what the cell and gene therapy or cell therapy clinical trial guidance refers to as the key safety issues, the ectopic tissue formation, biodistribution persistence, toxicity, tumorigenicity, and might be missing one there. But there's 5 of them listed in the clinical trial guidance. And those should be assessed in pivotal studies that meet this GLP, good lab practice standard. And facilities there should be certified. OECD members commit to certifying facilities to say they meet a minimum safety standard and standard for quality of data to make sure that the data is reliable. So these are the 3 stages you should be thinking about. There's no map to follow or necessary progression here or timeline, but I'll be talking about putting up a potential one at the end. And then the challenges that people have throughout preclinical studies, I'll summarize. This is what people come to me as. First of all, preclinical studies are expensive, especially GLP studies in special facilities. And so people have to make a trade-off between the costs that these take and the time it takes and the quality that they should be getting. So this is a bit of the 800-pound gorilla will push around and say, don't do these studies because we don't have the money now. But the problem is then you don't have the data. So it's a challenge that everybody has, and it's a reason why we have a bit of a valley of death. Oops, I jumped ahead. And then the second challenge that I find people have is they can't find and they don't know what is the relevant animal model. So this is a huge risk. So I think it's the white whale. Everybody's trying to find the best predictor of human experience for safety and efficacy, and it just doesn't yet exist. So it's hard to decide to invest in your studies, invest in your product until you know that the study that you're going to conduct, that costs a lot of money, will actually give you the results you need. But we won't know that until you have some interactions with the regulator and actually file a trial application to make sure that it's going to be acceptable and considered sufficient. And then finally, the preclinical challenge that people have is often irrelevant animal models are terrible predictors. This is the elephant in the room. We all know it, but there isn't a better solution yet. But these three challenges together really help to explain why it's so hard to navigate this valley of death, and why we need a bit of a shared learning so that we all determine how to best go about navigating translation to clinical trials. So, quick note, going back to our main plan for translation is getting our manufacturing to GMP. This is the reminder. You tell regulators how you manufacture consistent products under sanitary conditions, because that's the requirement under the Food and Drugs Act. And the regulations are written in a flexible way to allow you multiple ways to do this. And there is a role to engage regulators and ask them for feedback about how you're doing it. But I think it's a mistake to expect that the regulator will tell you how to do it, because it's going to differ based on the type of technology you have. It's going to differ based on the application space that you're in, and on business priorities and capabilities and your financial situation and your investors and all of these other things. Similarly, you tell regulators how your materials and preclinical models are relevant, how the package together stands to support a clinical trial application or a later stage clinical trial application or a new drug submission. And what the regulator does is tells you what their general expectations are, what the milestones are, and how to put this into a regulatory submission to communicate it to them. And finally, you decide how to meet good clinical practices and how you're going to assess the benefits against risks and how they shake out. And then you're going to present it to regulators. So when you think about this, there's no easy answers, but there is a process for going about doing this. And I would highlight, well, how do you align them? That is solved in part by this concept of quality by design. That's what it's trying to do. It's trying to make sure that you're advancing to get the right data package to put things together, knowing that multiple wheels are moving at the same time, and that you need the best data so you can figure out how those wheels fit together, and then figure out your design spaces if any of those wheels change. So that's what Panos is going to jump in and speak about now, and talk to us a bit about this from a development perspective and from the manufacturing perspective. So let me figure out how to mute myself now, and then I'll be moving ahead these slides for Panos. Where's my mute button? That's what my wife asks all the time. Where's my mute button? I don't have one.
I can mute you if you want.
Please do, and then I'll throw off my camera when I get a chance, but that'll allow us to move forward. And I'll pull up the full slides. Hopefully my slides are still working. Go ahead, Panos.
Thank you, sir. Thank you, Pat. Thank you. So before I begin, I think that it's very valuable for us to start looking into the therapeutic development as any product development. And for these purposes, I'd like to introduce a very simplified version of what a product development entails. So when somebody wants to develop a product, first of all, they come up with an idea. Right? This is the concept, as we say, and the idea always includes the final product in it. So we know what the product should look like at the end when it gets commercialized. Then the next stage in the product development pipeline focuses on demonstrating a feasibility. The parallel in the therapeutics development world, feasibility is demonstrated first in vitro in a lab setting, and then in a relevant animal model that will de-risk the application of the therapeutic on a human specimen. Of course, once the feasibility is demonstrated on any product, we move on to a more resource-intensive activities to develop a prototype. That prototype is what will then go to the final testing into the clinic in our case. And if the outcomes demonstrate a a proper safety profile and efficacy, then the product moves on to be launched for commercial release. Now, keeping these buckets at a high level in our minds allows us to demystify certain areas of therapeutic development. I'm moving on to the next slide. Um, what happens during the product development is that we ideally want to hit some milestones that will actually de-risk our investment both financially as well as effort-wise. So specific milestones in the therapeutics product development include, of course, the in vitro proof of concept, the animal proof of concept, and further down the line, the first in human application. However, in this graph, as you can see, the risk remains very high, even after the application in the first human. There are two main reasons why the risk remains high, even though the prototype product has already been applied on a human patient. And moving to the next slide, Pat, we can start describing, first of all, the nature of a product. The nature of a cell therapeutic product is very different than what most of us were used to in the biologics world. So our product has a very unique complexity to it. So cell therapies are very complex products that are impossible to characterize fully. Unlike the rest of the biologics products, it's impossible for us to take our drug substance, the cell therapy, and perform analytics to fully understand what they contain. We cannot, with 100% certainty, identify everything that happens within the cell. This is one of the biggest challenges during the product development, because that means that it adds an extra challenge to demonstrating comparability as we keep on evolving the manufacturing processes during the product development. Moving on to the next slide, I want to touch on the second reason why the risk of our investment remains high, even though a product can reach the clinic. Now, the second reason is focused mainly on the approach that the investors and the developers have decided to follow so far in the cell and gene therapies. And what I mean by this is that we have focused our efforts mainly on bringing a product to the clinic. However, as we all know, bringing a product to the clinic should not be our final goal. Our final goal should be to bring a product to all our patients, to bring a product to the market. So far, the milestone of the IND acceptance is the golden ticket, as we call it. And unfortunately, that leaves, if we move to the next slide, Pat, that leaves the product with certain weaknesses once it goes to the clinic. That means that when we go into the clinic with a product that is designed to hit the IND acceptance as a final goal, we unfortunately end up transferring a product that we don't understand fully, a product that is manufactured based on a non-scalable process with no proper qualified analytics and research grade raw materials. Now, why is this a big bottleneck for the long-term development of the product? Given that the regulators allow for such processes and such products go to the clinic. These are big bottlenecks because they increase dramatically the risk for clinical holds during the clinical phase of product development. Why do they increase the risk of clinical holds? Is because any big change, any change that needs to take place in order to bring the processes to a level that will be able to support later stages of the product development would have to be accompanied by data sets that demonstrate comparability. And as we mentioned earlier, it's extremely challenging to demonstrate comparability of such a complex drug substance. Therefore, there is increased risk of the developers having to go back into the development timeline all the way to the preclinical studies and repeat preclinical studies with the newly established manufacturing process in order to convince the regulators that the drug substance is actually equivalent and comparable to the drug substance that had generated the original preclinical data. Moving on to the next slide, we have a strategy that we believe in and we believe that can actually deliver the quality that is required in the most efficient possible way. This strategy is based on the quality by design principles. And these principles start with a single thing that we need to keep in mind, and that is starting with the final product in mind. Final product is the product that reaches the market, not the product that goes in the clinic. As we described earlier, the product that goes into the clinic is actually the prototype. Now, aiming to go into the clinic with an in-depth understanding of a scalable manufacturing process supported by qualified analytics and using GMP grade raw materials allows us to demonstrate comparability a lot easier. So following a strategic approach that will front load our development efforts to bring a product that is more understood and produced by a scalable process into the clinic will serve us very well in the long run. Now, I want to break this apart in the next slide. And I want to use an analogy that Pat had shared with me a few years back, and ever since he's allowed me to use. And this is the pie analogy I like to use. Now, I use the pie as an example of a product to demonstrate how simple in principle all the steps we need to take are. So if somebody wants to develop a pie as a product, the first thing that they need to think about is the target final product. We need to think about what kind of a pie we want to bring into the market. Now, that pie could be a cherry pie, an apple pie, you choose, but if you want to develop that as a product, you have a clear picture in your mind about what that should look, taste, and feel like. Now, the next step that we need to take in order to bring that product into the market is to come up with a recipe, come up with a procedure, the manufacturing process that will allow us to hit that profile, hit that product profile. Then moving on, the recipe itself needs to come up with specific ingredients, specific instructions, key instructions in the recipe, as well as equipment that are necessary for us to actually build this product to the quality level that we desire. Now, all these elements are the inputs of the product development. Now, on the other side of the spectrum, we have the outputs, and as outputs, we call the qualities of the product. Now, these qualities can be intermediate quality attributes, such as the nature of the crust that we bake, the filling consistency, the syrup viscosity. These are intermediate quality attributes. And then there are the final quality attributes that speak to the qualities of the actual product. Now, to me, when I first thought about this analogy, it was a bit shocking about how well it overlaps with what we need to do in the actual therapeutics product development. So if we move to the next slide, we're going to see that same way as we started with the pie, we need to develop a target product profile for our therapeutic. We need to know what the indication is, what the dose will be, what's the market we're going to target, and all the rest. So we have a very specific target product profile in our minds. Now, that doesn't mean that that target product profile won't change during the product development, but we need to have a target in mind that we're aiming. Then, similarly to the recipe of the pie, we have to come up to develop a process flow diagram. This is actually the recipe of the manufacturing of the actual product. Now, that recipe needs to include, of course, the material attributes. What are we using during this recipe? It needs to include key process parameters. We're talking about the important steps in the manufacturing process that can have an impact in your qualities. And of course, we need to identify and qualify the critical equipment that are necessary for manufacturing of our process. So moving to the other side of the spectrum, similarly to the pie, we also need to have a list of our qualities, the intermediate quality attributes, which can be I'm just providing some examples now. It could be for the expansion, it could be the phenotype of our cell population, it could be the genome editing efficiencies. So these are the intermediate quality attributes. And then we need to also define our quality target product profile, which includes all the final critical qualities of your product. These qualities speak to the purity, the dose, the potency, safety profile, and identity of your product. So you see that there are a lot of parallels between any product development and the therapeutic product development. And I strongly believe that bringing into the discussion other kind of product allows us to properly deconvolute all the steps that we need to take in order to develop a therapeutic and bring it to the clinic and beyond. So moving on to the next slide. Once we have identified all those puzzle pieces, one can now start building a what we call correlation network. In simple words, what we need to do is we need to define how any of these inputs of the potential critical material attributes, process parameters, or critical equipment can impact any of the outputs, any of the qualities. With such an in-depth understanding of the manufacturing process and the qualities, demonstrating comparability over the long run is becoming a much more straightforward exercise, because now we can really prove how any changes in our inputs, any changes in our processes and our materials can affect the qualities of our product. Therefore, demonstrate comparability as we make the slight changes that we will have to make during the clinical development of our product. So, having spoken a lot about pies, I would like to bring us back to our therapeutics development and speak a little bit more on a case study example that I put together in order to get a better understanding as to how these principles apply. So the whole QBD-based development can be split into two phases. The first phase, I'm going to start describing right away, is focused on an in silico effort, in silico body of work, or the preparation phase, like saying. And the second phase is the experimentation phase. Now, once we have the target product profile, we understand what the therapeutic we want to develop is, we then need to, as we described earlier, we need to draw the process flow diagram. Here I'm presenting a process flow diagram of a hypothetical therapeutic product, which is an iPSC derived allogeneic product. So you design the flow diagram and break it down to the smallest and highest resolution unit operations. Next, what we need to do in order to build quality into our manufacturing process is to subject each unit operation to the exercise that I described previously, which is breaking the unit operation into all the possible inputs and identify all the possible outputs. As we move on, and can we move on to the next slide, Pat? I want to pick one of the unit operations to dive deeper and walk you through the whole exercise that we need to follow. So for these purposes, I always like picking the unit operation of cryopreservation. And I do so because cryopreservation is actually one of the simple unit operations, yet can hold the keys to a successful product. It's usually overlooked because it's sitting on the end of the manufacturing process. However, the systematic deconvolution of this unit operation will demonstrate to you how complex it can actually be once you peek under the hood. So moving on to the next slide, once we have the cryopreservation as the unit operation, we can move on to the next slide, Pat. So cryopreservation as a unit operation can be broken down to even higher resolution steps of the process. It can be broken into the pre-freeze harvest step, the formulation stage, and then the freezing stage. Now, each of these stages can be broken down into inputs and outputs. And I like using the CMC acronym to keep me true to all the nomenclature that we need to follow in order to speak the same language with our regulators. So on the input side of things, we have our chemistry, and our chemistry describes actually the reagents, consumables, and equipment, and our manufacturing, which is actually the manufacturing process, the process parameters. These are all our inputs for each of these subunit operations. And then you have the outputs, which are all the analytics that you need to develop in order to measure the quality of this unit operation. Now, I want to demonstrate to you how complicated a simple unit operation like the cryopreservation can be and should be in order for us to ensure that we understand our processes and that we won't have any issues demonstrating comparability as we move forward. Moving on to the next slide and focusing a bit on the pre-freeze harvest subunit operation. There we go. Thank you, Pat. Focusing on the inputs of the pre-freeze harvest stage of the cryopreservation, we can see that under chemistry, we can very quickly identify the dissociation reagent as well as the dissociation stop solution being two of the reagents that can have an impact on our qualities. Now, if we zoom in further, we're going to see that there are a lot of attributes in these of these reagents that we need to take into account, and we need to deconvolute and put on our map. Then moving to the manufacturing processing, we see that the dissociation process as well as the retrieval can be two key steps in deconvoluting this manufacturing unit operation. And on the back end, on our outputs, we can see that the short-term cell viability or long-term cell viability, cell numbers, osmolarity, and other quality attributes are qualities, the intermediate quality attributes that we need to focus our analytics on in order to demonstrate that the manufacturing of this unit operation maintains a high quality. Now, similarly to the pre-freeze harvest, if we move to the next slide, we can see that with the same principles, we can deconvolute and break apart into all the possible input pieces and output pieces, the formulation of our material, And then the same way we can also deconvolute the next slide, the freezing step of the cryopreservation. So we have now built the whole map of all the pieces, all the puzzle pieces, the inputs and the outputs of this unit operation. Now, how do we move on next? And how do we build what we call a correlation network in the next slide? So we have described all the inputs, all outputs, and now we have to start somehow creating this correlation network. I try to identify, try to build some understanding about how all these inputs can impact our outputs. And this is where we will start being more creative in our ways. And this is necessary because, as we saw, the variables that are introduced in the system are a lot. There is a vast number of variables that we can experiment on to define this correlation matrix. And of course, their interactions can also exponentially grow the number of factors that one would have to focus on experimentally. Now, in a startup environment with limited resources and time, we must somehow prioritize some of those factors to experiment on. We cannot tackle everything. It's impossible. The solution space is too big for us to try and address every single interaction that is there. Now, the most systematic way to reduce the required experimental conditions start with a thorough risk assessment, which is then followed by the experimentation, a DOE-based interrogation of our system. So in the next slide, I'm going to see an example of a risk assessment output. And what is a risk assessment? Risk assessment is just a structured and systematic approach to identifying and understanding the potential risk or impact of the inputs onto the defined outputs. The result of the risk assessment is just an assignment of an actual number, what we call the risk priority number. And that number prioritizes the factors on a scale based on the impact that they can have on the qualities of your product or your unit operation. Now, based on the experimental throughput and resources of the R&D lab, the first screening experiments are designed around a short list of the top scoring inputs, as they have the highest probability to affect your outputs, the intermediate quality attributes or CQAs. Now, moving on to the next slide. If we can move on all the animations, but thank you. So the first thing that we need to do before we step into the phase 2, which is the experimentation phase, is for us to build our senses, build our analytics. It's extremely important for us to start there, because without our analytics, we literally cannot trust any experiments that we perform. At minimum, in order for us to start interrogating our manufacturing process, we need to have our analytics built to a tier 1. Tier 1 includes, of course, knowing the chemistry of your assay. So that will include all your materials, reagents necessary for your assay. And then we need to have a defined process for the assay. That's the SOP. Then we need to define the specificity. We need to ensure that the assay detects and only detects the desired molecule. Then we need to define the accuracy and ensure that the concentrations, amounts of what we're measuring are accurate. And very importantly, we need to have an idea of the repeatability of the assay. Can we trust our measurements throughout time? Can we trust our measurements if they run now versus a week later? So this is the minimum requirements for an assay to be trustworthy for us to properly deconvolute and translate the experiment outcomes that we get from the lab. Then, of course, we can move on to bringing our assays to a tier 2, at which stage we're preparing our analytics to go into the clinic. At that stage, we're defining our limit of detection, quantification, the linearity of the assay, and of course, we want to get an idea about some intermediate precision, which refers to the reproducibility of the assay when it's performed by multiple different operators. And then at the end, once we step into the clinic and we continue developing our product and analytics, we focus on the robustness of our analytics and we build in some redundancy of materials and we identify also process stability. Now, once we have our analytics, we can move on to performing our experiments. So the quality by design principles are based on a strategic and systematic approach to breaking apart any problem, any system, any manufacturing, using a series and iterative DOE experimentation. So building an understanding and control by constructing a correlation network between all inputs and outputs is established and is also supported by the FDA using a DOE approach. So this framework can fully describe the relationship between inputs and outputs, can help us identify how every variable in a manufacturing process can affect our qualities, intermediate or final. So 3 are the stages of the DOE experiment approach. The first stage is focused on understanding the system, and it's an approach that is sitting earlier in the product development, focused on research. The second tier is focused on optimizing your manufacturing, and the third tier is focused on characterizing your manufacturing process. So diving deeper into these 3 steps of the DOE deployment, and moving on to the next slide, the first stage or the research stage of the DOE deployment. But if we can move on through the animations, thank you very much. During the research phase, we build understanding. And in order to build understanding, we need to figure out the significance of the factors that we have chosen to screen on the qualities. So we want to see if we move the lever up or down, if we change the concentration of one molecule in our media, would that have an impact on the qualities we have chosen to measure with our analytics? Now, the screening designs, DOE screening designs, there are multiple approaches. There are fractional factorial designs and a lot of other thoroughly established approaches actually through other fields that can help you define the significance of all the factors. And not only that, but they can also help us define the directionality of the impact. In the next slide, once we established the significance, then the same designs with the same iterations provide us with an understanding of whether the impact moves the qualities up or down based on our manipulation of our systems. So through this phase, we get a good understanding of our process. We know now whether a process parameter or a material attribute are actually critical or not by demonstrating the impact they have on our intermediate quality attributes on the back end. The second phase of the DOE deployment and the optimization phase. In the next slide, we then focus on defining the exact optimum solution, defining the combination of inputs that will give us the best outcome on our intermediate quality attributes or the final quality attributes. This is the phase of the optimization. This is the process development phase, and it can be applied only after you have zeroed in and you have reduced your solution space to a handful of factors. This is a high resolution approach, and it cannot be applied on a very wide solution space because that would require thousands and thousands literally of experiments. And the final stage of the DOE deployment, and the next slide, we actually perform a process verification, and we define what is called the design space. The design space indicates what are the borders, what are the limits of a manufacturing process. What is the set point we know through our SOPs, but then for any deviation that may occur during manufacturing, we need to be sure, we need to have a predictive power to be sure that the qualities will not be affected. So at this stage of the process characterization, we define those borders by demonstrating what is the operating range, the acceptable range in the characterization. And with this, I'll pass it on to Pat. I hope I haven't confused everybody, but should you need any support with this approach, the quality by design approach to product development, there are experts out there that can definitely help guide you through these efforts. It seems complicated, but in reality, it's all based on simple concepts of product development.
Thank you very much, Panos. I am going to take it back. I have a final slide that I'll go through, but I first want to acknowledge now I'm hungry. You used the pie analogy, and it's around lunchtime. No, just kidding. But I think the pie analogy I really enjoy. It simplifies things for people like me, but I love when we have people like Panos who are technical experts who can get into some of the details and really help to navigate those steps. So I'm going to do a final slide here, just highlighting how these things can overlap in time and to bring it to a much higher level. So here, what you see is a very high level timeline, years 1, 2, and 3, that starts with proof of concept and initial plan according to quality by design. The dates I put in here are placeholders. I've seen things navigate to first in human as short as about 14 months. And sometimes it can take up to 10 years. So there's a huge variety here, but I wanted to put something in to actually guide and make us aspire to a reasonable goal. And that's what I'll be showing. So at a high level, the things that Panos was talking about, the process and the analytical development efforts should be starting once you've selected which product you want to move ahead. And they start off pretty light and then get really into the details that Panos was going through. And at the same time, you should be starting to assess your product and learn about it from a safety and efficacy perspective. So as you're developing analytics and measuring things and developing those tools to increase your confidence in them, you should also be using the product you're manufacturing and the different materials you use and the different processes as you're selecting relevant preclinical models. This includes in vitro and in vivo studies. So there's some model selection work here. And then you should be doing a bit of a pilot study lead in to de-risk high-level GLP studies that I was talking about before you align the two. By the time you get to pivotal safety studies, you should be using a product that is as close to your clinical product as you possibly could be. Ideally, it's manufactured under full GMP conditions using GMP materials and scalable process. You should have a prospective strategy for how you're going to demonstrate comparability between different product versions to draw into relevance things that you've worked on before. You should be doing those preclinical studies leading right up to your first in human study, and you should have a robust study design that will confirm some of your expected benefits and that some of the potential safety hazards are avoided. Now, you'll probably say, hey, Pat, how do we align our regulatory activities around this? And I would say you should be using them as tools to de-risk large investments. Therefore, you should be thinking, if you get 2 kicks at the can, your first pre-submission, in Canada it's a pre-CTA, in the US it's called an interact meeting, should be used to inform which materials you're going to use and to make sure that you're going to be considered sufficiently GMP compliant to get into your first in human studies, so that you're going to do your pivotal studies, your key safety data studies, using a relevant representation of what your product will be in humans. You should also be looking towards making sure that your animal model that you plan to establish the safety of the product will be done in a GLP facility, and you should discuss that with the regulator. And then eventually you're going to start doing those preclinical studies, and partway through that, when you have your interim readout, if you want the most efficient path to the clinic, you should be thinking about having a second regulatory interaction to de-risk your clinical trial design, to make sure that you'll have enough safety data and promising efficacy to get your clinical trial application approved, and to know where to put all of the data in a regulatory submission. So that's what your second pre-CTA in Canada or a pre-IND meeting can be used for. And then finally, you're going to be preparing a clinical trial application in Canada or an IND in the US. And you should be thinking about how you're going to align the activities of those 2 things. In this example here, I just put, imagine that you prepare a Canada first clinical trial application, and then convert that to an American IND if you wanted to go there. But all of these decisions, there's no right or wrong answer. You're just thinking about in terms of implications and putting together a strategic approach. So that's what I wanted to bring us back to say these things, all these activities, they're not just a nebulous philosophical concept. There's actually a timeline and projects and money you should plan to invest. And this is the story you'll need to tell investors about what you're going to do. And eventually you're going to show in hindsight, this is what we did and why we did it, to regulators. And then they're going to review it as a third party that's independent, that's focused on safety, quality, and efficacy. And then ideally, you'll get into your first in human studies and then do the same thing for development paths beyond that. So the key here that we're talking about right now, bringing back to a learning objective, is how do we understand our GMP, getting our manufacturing to GMP? How do we understand our product safety and efficacy, getting our product to GLP? And how do we align the 2? So with that, I think I'm wrapping up. I think we've left enough time for questions, and I think we have about 25 minutes for that. So we really do look forward to the discussion now. And thank you, Megan. It looks like you'll be there to help us navigate some of the chat questions and how people can ask us questions. And I wanted to highlight that we are reachable. Any interactions best start with questions. So if anybody is compelled by some of the things we said and really wants to know how it applies to their product, we're available for questions now. And over the next little while, I think you'll be able to get our contact information online. So Megan, I guess I'll turn it over to you. And I don't know, do you want our panelists to keep our cameras and mics on, or do we just pop on when you ask us questions?
No, that might become a little too pokeroo. So I invite Panos to turn his camera back on, and Callie too, if you're planning on joining us, we welcome you to join in at this point as well. So first of all, thank you guys for a really, I think, compelling discussion. You put it certainly in a way that as a non-engineer, at the very least, and a non-biomanufacturer, I understood DOE a little bit better than I did before this. So that's a good start. And so with that, I'll segue to the first question, which this person says first, thanks for a wonderful presentation. This question is for you, Panos, and it's about cryopreservation. So the person says, this is a very timely example as they're undergoing a process right now for cryopreservation. We have identified potential variables and preliminary data we can use to determine their significance and criticality. As we move into DOE, where can we go for more info on these steps, and what software can we use to determine efficiency, etc.? And they also point out that they're not engineers. So I'll leave it with you on that one. And then I'd like to actually maybe add on to that question, and that would be, what is the applicability? And I guess I know you say there's, no right or wrong, but this kind of segues into the next question, and I'll come back to that momentarily. But are there any considerations for academic versus non-academic groups that would be looking to do this? So over to you, Panos.
Yeah, that's a great question again. First of all, I need to emphasize on the fact that the DOE approaches are the cheapest, most efficient way to get to your answers, no matter what the question is. It does sound a bit complicated, but in reality, it's something that has been explored for decades in different fields. So I really encourage everybody to dive into these approaches as early as possible. So if I were to talk about academia versus industry, I would say that academia actually would benefit a lot more from the DOE approaches, given the more limited resources there. To come back to the original question, some softwares that somebody can explore, GMP is a very powerful statistical software, a little bit more complicated. Minitab is a very user-friendly platform. And as per advice, I would point people towards a consultant. It won't take a lot of time for somebody to teach a champion within their academic group or the industrial group that can then take the baton and lead the teams into a DOE deployment within the teams.
And can I jump in for a quick second? Because I see Panos answered both questions, but the one nuance that was added to the other question was also about how realistic is it for academia, and do they need to use CDMOs and those? So that whole section, I'm just going to comment from a regulatory perspective. There's no specific requirement that you need to do those things. And there's no difference for regulatory requirements for academia to industry. And I'm going to go out on a ledge a little bit and say, and there shouldn't be. We should be a little concerned if there was different thresholds, and I'm sure there would be a lot of discussion and challenges if there were different thresholds of safety and efficacy based on who's submitting this trial. Now, there are some differences in how they might be treated. You might want to expect our government could provide some more feedback to academic researchers who are doing this. Now, that's not my experience so far, and I'm not putting any value judgment on it, but the point is that we want our products to be manufactured under safe conditions in a consistent manner. And so whether you use a CDMO or not, the question becomes, how consistently can you manufacture the product? Which goes down to an evaluation against good manufacturing practice principles, which are necessary and by requirement in Division 5 and Division 2, and the link between them are required. Anybody in clinical trials must say they meet principles of good manufacturing practices. The flexibility in the interpretation is the tricky part, but you will not have to use a CDMO. It just so happens that they're expected and should have quality systems in place. They should have people trained to do things consistently. They should have environmental controls that are necessary for GMP facilities. So it comes down to how GMP-like are they, and can they do those things? So that's from a regulatory perspective. And I don't know if you want to add anything, Callie or Panos, on any recommendations there, but that's just the playing field. We're talking about quality of data and product.
And Callie, perhaps if you did want to weigh on this question, I haven't introduced you, so perhaps you can introduce yourself.
Sure, no problem. I'm a co-founder and director in weCANdev together with Panos and Patrick. My area of expertise is more on the stem cell side and non-viral gene editing, and work a little bit more closely with startups as they translate their concepts into early process development. I think the only point I really want to add here is that I frequently think of my academic career, and had I had the tools and the knowledge to use quality by design earlier on and DOE-based approaches, I would have flown, and being able to go back and now speak to others about implementing it. It's incredibly valuable. Consider it, and definitely reach out if you need additional guidance there.
Thanks all three for that. And I would just add in weCANreg, for example, pays for that valley of death biomanufacturing, and that would include DOE experiments, So I think there are opportunities to support this type of work with academic groups or within academic groups. And then the only other comment I would make to this is that CDMOs can be in an academic institution or be in those types of research institutes. And so I would say start early. So this is something we'll be talking a little bit more about next next session, but there are engagements that you can do with early phase biomanufacturing facilities that might have, you know, if it's coming to a cost, may have you know, opportunities to develop grants together and things like that. But I'll pause that thought for next week. So again, I welcome everyone to answer or to ask questions in the chat box. With that being said, though, I'm going to turn over while we're waiting to ask kind of a generic question, but one that I think gets asked a lot. And that is, where do you all see the biggest CMC gaps, for instance, for people bringing their biotherapeutic forward for the first time? And so I should specify gaps in knowledge, perhaps, but also gaps in other elements as well.
I can jump on it and then pass the baton to the two of you guys. My opinion is that the biggest gap is in the understanding of the process. Therapeutic developers bring a manufacturing process, a protocol that delivers the product in a black box, and the understanding of how any input can have an impact on your quality, on your product quality, is not there at all, and that immediately leaves the product with a very, very high risk profile. And unfortunately, I personally have seen therapeutics that could easily make it to the market if we had the luxury to go back in time and design the therapeutic development appropriately. It's sad seeing that. So I would say that, in my opinion, at least, the biggest gap is in the understanding of the manufacturing process. I'll pass it on to Callie or Pat.
Okay, yeah, thanks, Panos. Definitely picking on that point. I think underlying that lack of understanding is often insufficient planning leading through your development efforts, and then this urgency that Panos touched on to get to first in human as quickly as possible. So those two drivers often lead to that, and it really takes a concerted effort up front to say, no, we wanted to devote time and effort to these development studies to build our understanding so that we don't get tied up and we're not at risk of being put on hold. So it's very much a shift in mindset, but once implemented, should lead to a lot less pain.
And I have the benefit of going last, and I'm going to build on it. I'm going to say I agree. This is the biggest gap that I see, and I just say it's a bit of a gap in understanding, but it's a shared understanding. This is a challenge. We talk about and you hear about quality culture and understanding that it's not just about how and what to do, but how and what to do together and being on the same page. And I think you see some companies where there's a wide range of understanding. There's people who are often more on the research and development side, and then you have people who are right on the GMP side, and everybody in between. And that range is a challenge, and you need to get everybody to understand where they need to be when on this quality culture. And a very important role is the glue that sticks together and lays that quality culture, and people like process and analytical development roles, the MSATs in a team, the tech transfer, these things, they're really difficult to do. And so getting people on the same page to do it is a big challenge.
Any tips for doing it?
Have a clear strategy, aligned to quality by design. Sometimes it does help to get someone in who understands it really well and can help put the plan together. Orient the team, get on the same page, and then you can start adopting it. There are some tools. There's a question earlier about tools, and I wanted to highlight there are software surrounding the quality by design. QBD Vision, wonderful tool. Things like this can help. And then the other part is making sure that you have good orientation, strong plans. And we're obviously very biased towards quality by design. We have between us some early papers on it. We have applying it to cell and gene therapy. We have the experience of applying it. So get roles like that who are able to internalize it for a company. And if you don't have it, then look externally for a short time to augment your own team and accelerate, right? Because this takes time. You might take 6 months to a year and a half to find the right people to work through and understand how to think about your product through a quality by design lens, and to get them all rowing in the same direction without hitting their oars together. So the quicker you do this and the more intelligently and strategically you do it, will lead to more success. And it should give investors more confidence that they're investing in something that could work. Because we have lots of, Panos mentioned it earlier, you might find a product that might work perfectly, but it doesn't have the right plan and you don't get the right data. And even if you do, it's not the right quality. All these things have to align. It doesn't just happen. It's not magic. It's translation. It's intelligent. It's strategic. It's all of these things.
And keep in mind that the target is the product, not the prototype. Don't focus on getting to the clinic. That's not enough. That's not good enough. We have the responsibility of taking products to the market, not to the clinic. And that would be my 2 cents.
Great, thank you. So maybe as a housekeeping note, Patrick, I missed the software that you had said. Panos, you had said GMP and Minitab. Do you mind writing those just in the chat box, just to make sure there's no confusion in which programs you're referring to? So as you're multitasking doing that, I'll continue with questions. No pressure here. But are there any gene therapy specific resources or expertise that can be accessed by academic groups for managing the early preclinical translational steps? So I'm going to take a first crack at that, just to say that weCANreg has a 5-day in-depth hands-on in-person workshop that brings in subject matter experts. It's called Preclinical Program Development Workshop. We are looking to offer this again in the new fiscal year, pending some funding decisions. But so keep your eyes out for that, to the person who wrote that. And there are other, I think, opportunities coming up too, that Stem Cell Network and we and perhaps NRC are looking to develop out from this session. So the idea being to turn this into another in-depth workshop. So stay tuned for those as we develop them out. But over to you guys if there's any other resources you would recommend.
I'm having trouble typing it. I can type to hosts and panelists or individuals, but I can't put to everybody right now. So
yeah, that's right.
Yeah, so QBD Vision is a group that we like working with. The tool is helping people think about these steps and make sure that you put in data there. So it's a very broad, useful tool was what I was referring to. And now your question, and now my brain was on the tool. I'm sorry. So one of the other guys can answer.
I know.
I'll see if I have anything to add.
What a moderator thing to do is ask you to multitask and ask a question at the same time. Are there any resources outside of those, but just any tools or trainings that you're aware of that would help specifically, I think, academic groups managing these early preclinical translational steps? And I will say this is a very big gap in the academic world, is taking that first biologic product that you have and starting from an academic group perhaps, and then making your way through the translational uncertainties.
Maybe I can start, and then Panos and Kelly if I miss things. But I wanted to highlight the weCAN entities, weCANtrial, weCANreg, weCANdev, and the weCANtranslate network has a library of materials that we rely on, that we develop. So I offer it, and I'm not going to go into the details right now, but with some of the partners you mentioned, I mean, we do a regulatory literacy program with Stem Cell Network that can go to people who are part of the network and are grad students working in those labs that are awarded. I love doing that so people can get tools through that. They can reach out to us to work through things. I have training that I do from a regulatory perspective through some of the other NCEs. We have an entire companies in a box ready to go. And I don't think we'd be the only consultants to do it. I think we're just the only ones focused on cell and gene therapy who have the experience that we do. So I'm plugging ourselves a little bit in that we have tons of materials. And I think I lecture at 15 universities in Canada providing these all the time. We work through OBio, we work through all sorts of different groups to provide these materials. They're available. And people should reach out. And I think training is wonderful and I think extremely important. And I'm really happy to see weCANreg, Stem Cell Network, and these groups stepping up in that space. And we want to be good partners in it. So let us know how we can contribute. We have it all. I have hundreds of presentations, just canned presentations ready to go.
Thank you for that. And I invite, Panos, yes, go ahead if you wanted to add on.
No, I was about to say that unfortunately there is a lack of tested tools out there. It took me and Kali years and years of very focused efforts trying to build a strategy, trying to build our approaches. So my advice would be to bring in people that understand the field, bring in people with actual hands-on experience, and be overly critical. Don't trust them because of a name. Don't trust them just because of the background. Make sure you ask questions, or make sure you understand everything that somebody is advising you to do.
Fantastic. Thanks so much for that. Okay, great. And I'd say when it comes to that, when it comes to outsourcing or consulting, so what do you look for in finding a partner, finding a consultant? What matters most to you?
Who wants to jump on that first? Callie, do you want to jump on that first? What should people think about when they reach out to consultants?
They should come with specific questions, areas that they're interested in tackling first. They should absolutely look to see whether or not their consultants have expertise in the space, if they're able to communicate on what their gaps are. So they're able to draw the connection to where they are now, identify the gaps, and bring the group along with them. You should make sure you understand what your consultants are saying and that it's not all a foreign language to you. It's incredibly important that you understand, you agree with the direction that you're going in. So finding the right partner for that is critical. And then growth, I would say, is the other one. So not just for the now, but for the future, who you can stick with for that integration across development, regulatory, trial, for example, as Pat showed. So you have that glue, and yeah, they can help bring you across the finish line for some of those critical inflection points.
Panos, do you want to go next? Do you have anything to add? What would you look for? You agree with Callie?
I'm covered, and I would emphasize on make sure you understand what they say. If they cannot break it apart into simple pieces, that means that they don't understand. So ensure that you understand every single step they're proposing.
I love that, and I'll agree with both Callie and Panos. And I just want to add, I would never sign a blank check. I think you should understand what things you're going to get and how you're going to get there. I see some people signing like for hourly rates. That's not the most important thing. The most important thing is that you know what you're going to get and how long it should take. But the things that you are handed are actually quite helpful, and having somebody walk through that with you and show you where they think you need to go, how they're planning on getting there, and shows you a bit of what it looks like. That's the best way to start a conversation. I think there are people in the space who have well, actually, they're all at different levels of experience, but can't say exactly what you'll get and just want to give you the right answers. And I would be skeptical of working with someone who puts it that way, because first of all, there are no right answers. You must think in terms of implications, which means that the group that you're working with needs to explain what they're doing and why, and you should be able to understand it. If not, that's a red flag for me. And then what it is they're going to do and how they're going to get you to a value inflection point, how they're going to move the needle for you, that should be clear when you engage with a consultant in the space. And that kind of direction is very valuable. And if somebody can't communicate it to you, then there's not a match yet. So that's very important in my mind, and much more important than just cost. I think that's a very big driver for people in this space, and we have to, I think, not let that be the gorilla, the 800-pound gorilla in the room. It's easy to turn to somebody who's had a success in supporting one clinical trial, and that may be not too expensive, but that might not be the right partner, and they might steer you awry with the experience they've had. It's different than yours. So you need, I think, the more experienced group that's able to speak intelligently to things and explain it to you.
Okay, thank you for that. There are two last questions that just came in. We've got about 5 minutes left, and I want to leave time for maybe final closing remarks. So that's 3 different things I'm asking of you, and then I need about a minute to close out. So rapid fire, maybe these last 2 questions. The first of which is, in terms of planning, quality control, and other critical aspects, how does the preparation for early phase clinical trials for RNA therapeutics differ?
Okay, keep Pat muted. They don't differ at all. The approaches are exactly the same, and that's the beauty of this approach. You can apply the same principles, the same approaches on any kind of therapeutic development. Cell therapies just happen to be a little bit more complicated. That's it.
Perfect. There we go. Okay, great. Next question. Great talk. Thank you both. For cell therapy specifically, what are your views on how the field should navigate the unknowns pertaining to genetic aberrations, mutations that may be detected in the drug product as a consequence of the evolving technologies? I mean, CRISPR obviously is a good example of.
I'll try and be
Pun
concise with
intended.
this one because I've been accused of being too chatty. But strategically, the FDA guidance that you get in this space is that we need to use orthogonal methods. No one method is best. So I've actually had the opportunity to speak to the FDA on a policy shaping committee to say, I think that people should think in terms of in silico assessments, in cell and cell-free assessments, and then also with preclinical studies. You should have a robust way of looking at this from a risk-based approach and say, look, we can have off-target effects, we can have unexpected cell functions, and we can have things that aren't really detectable and we can't even imagine, but we should do it in animal models as well. You need to think about it, and the best way is a process. It's risk-based approach. What are the potential hazards? How are you going to assess it with multiple assays, and understand what Panos went through before of what tier of assay are you using at what stage of development, and you need a strategic approach that aligns all of those things. This is a complicated question with a complicated answer when done properly.
Thumbs up for perfect. Fantastic. Very good rapid fire, guys. Thank you for that. So with that being said, maybe one final minute of question and answer, and that is to maybe all of you, and that's, what are your closing thoughts? What would you like to leave both our academic and industry stakeholders on the call today with from your respective positions?
I'll jump in. I'll start. So translation, as measured by you get into your first in human studies and get regulatory success, doesn't happen by magic, and it costs a lot of money. So do it strategically. Think about the simple idea of aligned to principles of quality by design. Manufacturing to GMP is a key milestone. Preclinical studies to GLP and what your relevant animal model is extremely important. And then you have to start thinking about the end in mind, like Panos said, and that's you're going to meet good clinical practices and develop a product that can be used in people over time. So don't be short-sighted. There are things worth investing in, and do it strategically. Inform yourself and try and use the tools around you, which may include consultants, or it may include making sure you hire the right glue in your team to bring these ideas together.
Yeah, I'll just jump in, and picking up on a point that I brought up earlier and has been throughout, make a plan, identify what you can do yourself, identify where you need help, and bring in the right people to do it. I think that's key, and be open to learning and adapting along the way.
And I'll close the discussion by saying that you just focus on the product. Understand that you're building a product like any other product that you see around you, like a coffee mug, follows the same steps. Don't overcomplicate things. Don't let anyone complicate things for you. Make sure you ask the right questions and bring in the right experts.
Thank you, Panos. I thought that was a missed opportunity to bring back the pie, which then I will do. So remember that pie. No, thank you all for your participation in this session. Patrick, you're never too chatty. I think we just are up on a timeline. So thank you all. I think thank you all for your willingness to engage with folks in the after this. As a reminder, this will be recorded and it will be on the Stem Cell Network's website very soon. The first one is already posted. Now, we hope that you'll join us for next week's session that's taking place on Monday, December 4th from noon to 1:30 p.m. Eastern time. And it's on when and how to engage with a CDMO. So I think some of the questions that were brought up at the beginning here will be really well suited for the conversation that's taking place next week. So with that being said, we have a few thank yous coming in. No questions, just commending a great topic and discussion. Thanks to the organizers, thanks to you guys, panelists. And I think it was really well received. So thanks for all of your time and energy, folks. And with that, we'll close it out, and everyone have a great day. Take care.
Thank you very much.
Thank you.
Thank you.